From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 14, recorded on January 11, 2021. I'm Vincent Racaniello, and you're listening to the podcast about the nervous system. Joining me today from New York, Ori Lieberman.
Hi, Vincent. How are you doing?
I'm all right. You're back on the East Coast, I see.
Yes, in the cold. Frigid cold.
What was it like in California? What were you, in the 60s or 70s even?
No, it was in like the high 50s, low 60s during the day, and then it gets cold at night. I mean, it felt like a little bit of a kind of a deserty feel, even though we were in Northern California. Yeah.
And from Salt Lake City, Jason Shepard.
Hi, Vincent. Hi, Ori.
Is it cold Jason. There?
It's cold today. We still don't see any snow on the horizon, which is kind of disappointing. Actually, last year was the driest year on record, apparently, so this is not good for us.
It's too bad. All right, this is your TWiN crew for tonight, but we can teach you some neuroscience. We have a cool paper. Who's doing this paper? Is it Jason?
No, it's me.
It's Ori. Actually, I was wondering if Ori had some inside information about this stuff because I don't know if you saw today, but Eli Lilly has a trial ongoing for Alzheimer's disease, and we can get into this amyloid hypothesis in this paper. But one of the big criticisms of this whole amyloid cascade is that these clinical trials have not been very successful. And just today, they announced phase 2 clinical trial data that was promising. Well, they announced it, but of course, we haven't seen any data.
Yeah, there's no data.
This is, I guess, apparently they have to announce it because it can reflect on the stock or something like that. But
the valuation, I mean, they raised like $20 billion or something today, or like their evaluation. I mean, they made a lot of money on a nothing statement. So.
Right. Yeah.
Alzheimer's is a big deal.
Money, so.
Well, okay, Ori.
Well, bummer.
What do we got here, Ori?
So this paper was actually less data than I was hoping. But I think what we can do is talk about Alzheimer's, as Jason mentioned, and we have a bunch of emails, so we can kind of go through this somewhat quickly. So this paper, which is called, let me pull it up, sorry, switch tabs now. Acute targeting of pre-amyloid seeds in transgenic mice reduces Alzheimer-like pathology later in life. It's in Nature Neuroscience. The first author is Ruth Ullmann, and then the last author is Matthias Eucker. And they're in Germany, in Tübingen. So this paper focuses on a kind of a fundamental question in drug development in Alzheimer's. So as many of you guys know, Alzheimer's is a disease, a dementia that affects a huge number of people in the United States and around the world. There's no effective treatments, and causes a significant amount of kind of healthcare costs and also suffering because the time from diagnosis to death can sometimes be 8 or 10 years. So you can have patients really have a tough time for a while. And over the last 25 years, there's been an explosion in research at the molecular and circuit level on what the pathophysiology is of Alzheimer's. And this has led to a number of clinical trials, which we kind of just talked about, we'll talk about again, that are really kind of targeted, designed therapies or rational drugs in some way that have mostly failed in clinical trials. And this paper tries to get at why they have failed. So let's take a step back and talk about what the pathology, or what is the thinking about what causes the pathology in Alzheimer's. So a lot of this has come from genetic studies that are actually really powerful, both in small families, for example, that have autosomal dominantly inherited Alzheimer's. So where you can really clearly correlate or generate some sort of causality between a genetic variant in family members that are affected and the absence of which would, or then the people in the same family that don't have the disease would not have this variant. And these variants come up in a set of proteins, including one called APP, or amyloid precursor protein. And this is a transmembrane protein that sits on endosomes. And in the brain, it's then cleaved into a number of different products, including a subfragment that's either 40 or 42 amino acids called A-beta, which is cleaved from the membrane, from the internal. It's internal in the endosome, so it's cleaved into the inside of the endosome, and then the endosome fuses with the plasma membrane and releases these A-beta monomers, or single A-beta fragments, into the extracellular space. And the A-beta monomers then can form higher order structures, so oligomers, and then later on longer fibrils that are high molecular weight and who have complicated structures that are really not relevant to go into details, that sit in the extracellular space and create some sort of pathology that is, again, not really understood, which leads to neuronal dysfunction. And a second neuropathologic finding where another cellular or neuronal protein called tau, which is
before we get into that, Ori, so one of the reasons why this genetics was so convincing was that the only way you can really definitively diagnose Alzheimer's, the way it's categorized, is by the pathology.
Oh, yeah.
And so we had known for 100 years that if you look at an Alzheimer's brain, there's two kinds of pathology. There's these amyloid plaques, which are extracellular, so outside the cell, and then the second pathology, which you're going to get into now, is these tangles, these neurofibrillary tangles that are intracellular, so inside the cell, and that's caused by a second kind of protein. But the fact that these genetic mutations caused changes in these proteins, the amyloid protein, and the peptide, the beta amyloid peptide, that was the main component of one of the pathological hallmarks of the disease, really was the most convincing, or still is the most convincing data, that if you target this beta amyloid and the plaques, that you would then hope to alleviate the disease.
Right. So the A-beta peptide that I mentioned before is the beta amyloid that Jason's mentioning, which then forms these plaques. And then these plaques are seen in parts of the brain that are affected and that are damaged in Alzheimer's. And then the second neuropathologic finding that Jason just mentioned are these neurofibrillary tangles, which are made of hyperphosphorylated tau, and tau is a microtubule-associated protein. And then once you see hyperphosphorylated tau, you can see neuronal cell death and synaptic loss and kind of general things that are bad when they happen in the brain.
Could I ask you how? So the pathology was originally studied on autopsy, but can you pick it up now by scans of some kind?
Right. So this is the hypothesis has been essentially a sequential hypothesis. And that's obviously so sequential, meaning that there's A-beta pathology or amyloid plaques, and then subsequently neurofibrillary tangles and that are made up of tau. And
so I think what Vincent's saying is, can we detect these in a person?
No, I know. So that autopsy data is cross-sectional. And what you can see is that, for example, time from diagnosis, you can see early from diagnosis, there's more A-beta and less tau. And then eventually you have more A-beta and more tau. And then now they're doing this in vivo with PET imaging, where there are tracers for A-beta, where you can longitudinally measure the burden of amyloid plaques in the brain. And then also tau, the hyperphosphorylation of tau.
But there's been a lot of controversy over which one of these pathologies is the most important. And one interesting finding that's sort of emerged over the last 10 years is that there's been a lot better characterization of dementia and cognitive deficits in people. And the correlation between the amount of these plaques, the amount of amyloid that people have, is actually not great with the actual cognitive deficits that people see. So, for example, you can sometimes find a brain full of amyloid, and the person cognitively was totally normal, and then vice versa. So, so there's controversy over the pathology, and the hypothesis that's emerged there is that the plaques are the end stage of the system trying to defend itself against producing this peptide, and actually sequesters the real toxic culprit, which are these oligomers that Ori mentioned, where if these oligomers are sequestered in the plaques, they can't actually damage the cells. But of course, you can imagine that once you've got this big physical barrier in the brain, that's obviously not going to help as well. But there's sort of this very, very long progression of the disease, and that's what we'll get into with this paper as well, where a lot of the clinical trials have sort of failed because it's been very difficult to diagnose someone who is going to get Alzheimer's unless they have this rare genetic variant. And so I would say the biggest challenge to the field is being able to give a therapy, no matter what it is, early enough in the progression of the disease, because by the time you have cells that are dying or dead, there's no way to come back from that. So you can maybe, halt it, but the damage is done. So, yeah.
So maybe I'll briefly talk about clinical trial design, I guess, or like how people think about clinical trials. So clinical trials are essentially ways to test interventions in humans. And these are generally very large enterprises that are very expensive. So when people design clinical trials, you want a couple things that make you able to interpret the results of the trial regardless of the outcome. So if it's, for example, an outcome that shows no effect, you want to know that, for example, the drug worked and that you're measuring something reliably. So in this case, the primary outcome in almost all of these trials is actual cognitive ability, which is in some ways a symptom and not necessarily a true objective measurement. So you could imagine that you could stop Alzheimer's pathology, so neuronal dysfunction or buildup of tau and neurofibrillary tangles or amyloid plaques, but not actually affect cognitive dysfunction if there are, for example, like circuit changes or other issues that affect cognition. And the second thing is to show that your drug itself is actually engaging its target. So a drug could also fail in a clinical trial if it doesn't actually inhibit the kinase that it's supposed to inhibit or clear the amyloid plaque that it's supposed to clear. So in a lot of these trials, what you'll see is the primary outcome be cognition or cognitive tests, but then the secondary outcome a lot of times will be these PET scans or blood tests or tests of the cerebrospinal fluid for amyloid levels to actually show that the drugs are having an intervention on this amyloid hypothesis and cascade.
And one of the issues with the trials, though, has been, who do you put in the trial? And, again, if you are exhibiting cognitive loss already, it's probably too late. So the newer trials have tried to figure out ways of, either putting in the sort of preclinical first signs of loss of cognition and then putting them in the trial, or now with these PET, the imaging where you can actually image the pathology, there's sort of better ways. But the problem with the PET imaging is that it's very expensive. Yeah, it's very expensive, and it's very it's not. I don't think it's that pleasant for people because you have to like inject this tracer into the blood, and there can be side effects and whatnot. But so something you probably won't talk about too much today is that there's a hunt for a biomarker, so something that you could just take a blood test and have that as a way to say, okay, you have higher levels of this protein or this marker in your blood, and that means that you're maybe going to get Alzheimer's, or it's puts you at high risk, so you should take this therapy now. Some kind of similar to prostate cancer and in men when you have levels of a certain protein that you could help gauge whether you're getting prostate cancer. So that's certainly, I think, something on the horizon that would really sort of in tandem with therapies that target the right thing would be great.
So the second issue with that is that not only is it difficult to pick your study population, but it's also hard to convince someone who's asymptomatic to enter into a study that involves a long follow-up and is kind of a burden on the patients when they might say, well, I may not even develop this disease in the first place. So some of these studies that are now ongoing are in populations that have genetic risk factors, which we won't talk about today, that are more prevalent in the population than mutations in APP or in the amyloid proteins, but have essentially are less certain that they'll cause Alzheimer's. And we can just kind of leave that to the side for right now. And
although
the reason
we
that it's
should
important,
mention, I mean, I often get asked this from the public when they take these tests like 23andMe and
like
whatnot.
23andMe, yeah.
And so, there's this common variant, ApoE4, that does predispose people to Alzheimer's dramatically, not as much as if you have a dominant mutation in APP or presenilin, but it does really enhance the risk factor. And I think what's emerging now is trying to figure out how this ApoE4 protein or this ApoE protein actually fits into the pathology. And I think that's a cool area of investigation.
And then there's also other variants that are really not expressed by neurons, but for example, are expressed by microglia, which are a type of innate immune cell in the brain. So you can have neuronal pathology or kind of neuronal circuit dysfunction from a non-neuron, which is also cool.
And the genetics are also actually pointing to that too, and that some of the less common variants, but highly, penetrant for the pathology, also are targeting these microglia cells.
So to take a step back, we now are talking about how we would design the trial. And just to kind of nail the point home, that we want to have asymptomatic patients or patients that are early in the pathology, because what's thought to really be pathogenic and cause the disease are these A-beta oligomers, or kind of pre-plaque species of A-beta that are likely present before you become cognitively impaired. They're present before you are positive on PET imaging. And what this paper will show is that they're actually likely present before you even see neuropathologically that there are amyloid plaques present. And so you could, what this paper essentially hypothesizes is that if you treat a patient with the drugs that we'll talk about in a second, very early, even before there is neuropathology, you could actually have long-standing effects that prevent this cascade from spiraling out of control. So what are these drugs that we keep mentioning? So they're antibodies. So these are this is a paradigm that you may have heard about in the news with the Regeneron antibodies for COVID. So this is called passive immunity. So you have an antibody that reacts against a pathogenic protein or molecule. So in the COVID case, against the virus itself, or in this case, an antibody against A-beta. And that when you infuse this antibody into the patient, it then leads to clearance or neutralization of this pathogenic species. And so there have been a number of these antibodies which have been developed by different companies, by Eli Lilly and by Pfizer, because as you can imagine, if you could make one of these drugs that work, it's a huge market. So there was a huge push from a number of different companies to make these antibodies. And
the history of this is kind of interesting because we kind of talked about this on a previous episode about using mice for translational studies, but in fact, this is one of the rare cases where the proof of concept, the proof of principle that you could use antibodies against a brain peptide were shown in mice and were successful. And there's mouse models of Alzheimer's, and they were cured by these antibodies. And so of course, we don't know yet if this really will work in humans, but at least to a lot of neuroscientists, there was this idea that this wouldn't work at all, because how could you get antibodies to clear out a peptide in the brain, which is supposed to be, immune privileged. There's supposedly no T cells. This is not something normal. And just the fact that that actually worked, and that they could show that you would, one, get rid of this peptide this way, and two, that actually reversed the pathology. So the direct hallmarks, the amyloid plaques, at the time was amazing breakthrough. And but one of the problems is that they went from that mouse observation breakthrough to clinical trials really quickly, like within 5 years, which is unheard of in. And then I guess this is again because the money here, of course, is that this is a huge
also the
population.
impact on people.
The impact on people, but from the pharmaceutical company's perspective,
they don't care about that.
The first company to sort of jump on this is going to make a lot of money. Let's be clear there. But they rushed these trials, and I don't know if you want to get into that a bit more. But they threw these antibodies into people. Sometimes the trials weren't even designed well to look at that endpoint. Some of the antibodies didn't even cross the blood-brain barrier. So you're like, well, of course it's not going to work. So that set back the field a lot because those clinical trials did not work. But they didn't work necessarily because the target is wrong. They weren't working for other reasons. So, yeah.
Right. So I guess that gets back to this question of do these trials fail because of a technical reason or a pharmacokinetic, pharmacodynamic, or an endpoint measurement reason? Or is it truly that the amyloid cascade and hypothesis is wrong? And I may get in trouble for saying this because I'm applying in neurology, but there's also a lot of personalities and money that is involved in this amyloid hypothesis, because the field has felt that this was really the driver of pathology for 25 years. And so there's it's hard to then say, well, maybe it's completely wrong and people have built careers on something that is wrong. I think that my takeaway from this paper and from reading about this field is that that's actually not the case, that we haven't really tested the hypothesis well enough. But that's clouding all of this kind of this discussion.
I would agree with that assessment for sure. There's the Alzheimer's field itself is full of. I mean, science is political. There are people in science that are human, of course, and so then they're going to want to, have their ideas being the one that's getting funded and whatnot. But so I'm also on the fence in that I think that there's some compelling reasons to say the amyloid hypothesis is correct. But putting all our eggs in one basket here is not a good strategy. And there's more and more funding now and emphasis on the tau side of things, partly because it looks like the amyloid cascade may not be wrong, but it could be that it's triggering the tau pathology, and the tau is actually the real bad culprit. And then there's questions about how you trigger all of this, and neuroinflammation is certainly something that's probably involved, but there's been these other ideas, like there's metals in the brain, there's some sort of environmental factors. But I think what's sort of nice to see in the basic science world is that now there is funding for alternative hypotheses, and I think this is going to be a good thing. But I also agree that I don't think the trials have really tested the amyloid cascade properly.
Yeah. So let's talk about this paper. I think that's a good I was trying to think whether there's anything that we should really cover, and I'll put a couple links in the notes for reviews that I thought were really good. There's one review from, I think, 2019 from David Holtzman's lab, who's at WashU, where they go through all of the different therapeutics that have been tested in clinical trials in Alzheimer's disease, and he has a really nice table talking about all the failures. But he gives a nice perspective because he's really an amyloid person. He's a person who's been a proponent of the amyloid hypothesis. So it's an interesting reckoning to kind of read this article. So let's talk about this paper. So if we accept that the antibodies are not working in clinical trials because it's a truly negative result. So giving these antibodies is doing something to A-beta, but it's not affecting cognition or cognitive ability, or neuropathology in other ways, so neuronal cell death or tau or anything like that. Then the question is whether, and the second part of that is that we know that A-beta does lead to pathology from the genetics and from the fact that it's present in these plaques. Then the question is maybe we're treating at the wrong time. And a problem with clinical trials is that because they're so expensive, you can't, ideally you would treat a population at age 40 and a population at age 60, and then you'd follow them for different amounts, and you could design the perfect experiments, but you're constrained by money. So what this paper addressed was whether you could treat subjects, in this case mice, and we'll talk about the mouse in a second, earlier in the course of the disease, before there's even any amyloid plaques, and prevent pathology. And so what they used is a mouse that harbors a mutation in the amyloid precursor protein that leads to amyloid pathology earlier, and then tau pathology, and then neuronal cell death. These mouse models this is one of many Alzheimer's mouse models. It's been very, very well characterized. So
by the way, fun fact, my first ever publication was on a mouse model of Alzheimer's. This was called the so-called triple transgenic mouse. And this was in Frank Lafeur's lab at UC Irvine, and I was an undergrad exchange student from New Zealand. And this was the first paper this was the first mouse model that had both amyloid pathology and tau pathology. And I was an author on that paper, but that was back in like 2003, so I'm dating myself there. And
the field hasn't progressed since then, so.
I know. Well, it's interesting because I did I did synaptic physiology on that mouse, and that was also at the time one of the first studies to do that. And but it's always interesting because that mouse model is being used so much that paper cited like 3,500 times or something crazy like that.
Wow, that's crazy.
The field's definitely use these models to the max. Anyway, carry on.
And what you'll see in figure 1A, which is I really think a nice figure about this mouse model, is that mice live to be anywhere between like 2 and 3 years old, let's say 2.5 years old, so 30 months. And what they show is that the first plaques that are seen with neuropathology are seen at around 9 months, whereas the first plaques that are seen by PET imaging, so essentially imaging of the live mouse, are not seen until 17 months. And that is really attributed to the sensitivity of the technique. But what they can do is that if they extract the levels of A-beta, so in this case, either the monomer, oligomer, or the plaque, using different biochemical techniques, you can actually see that the levels increase before the first A-beta plaques or amyloid plaques are seen in histology. So they show this figure where essentially by 6 months, you can start to see an increase in the level of A-beta. And even though they don't know what species this A-beta is, they hypothesize that if you could clear the A-beta at this time in the progression of pathology with one of these antibodies, you could then prevent the subsequent cascade of pathology and symptoms. And so the way that they test this hypothesis is really a tour de force. So they take, now I'm going to count, they take like 6 or 8 antibodies that are that have gone through clinical testing. And in clinical testing, of course, these antibodies are humanized. So they have human constant regions. And this lab made or collected mouse, or essentially they made antibodies with the mouse constant regions. So you can imagine that if you put a humanized antibody into a mouse, the mouse will have nothing to recognize the antibody and clear whatever the antibody is binding to. But in this case, they took a necessary step. They made them, I don't know what the equivalent of humanize is for mice, but mouseified the antibodies. And their assay was looking at the level of A-beta after infusion of these antibodies. And what they found was that only one antibody, so I guess they call it a chimeric mouse antibody, so CM, I wrote this down because it's a mouthful of what the antibody is called, and now I don't see it in my notes. It's
Crenezumab, but then there's solanezumab and
no, but the aducanumab, ADU. So adu is the one that they see has an effect on A-beta levels in the brain. And the way they do this experiment is they essentially give antibody intraperitoneally for 5 days, and then they wait a period of time, and then they sacrifice the mouse, and they do the extraction in the brain. So this is not an ongoing infusion of antibody. They essentially give an antibody bolus, and then they wait for clearance, and they measure the clearance. And this is kind of a side point that is interesting, is kind of the differences between the antibodies that they choose. So you can imagine that some antibodies might have affinity for an A-beta species that's already in the plaque. Some might have an affinity for A-beta when it's a monomer. Some might have affinity for A-beta when it's in an oligomeric state, or could have affinity for multiple different A-beta forms. The antibodies could also differ in their pharmacokinetics and pharmacodynamics. So you can imagine that this antibody has to be at a certain concentration in the brain. And if you give a dose of the antibody, but it's cleared rapidly in the blood, or it's sequestered in the blood and doesn't get to the brain, it may not reach that concentration for a long enough period of time for it to have its effect.
Do you know anyway, which company is this aducanumab coming from?
I think it's Pfizer.
Pfizer. I was quite struck by this as well, where that was the only one that seemed to have efficacy in this assay. The one that we're talking about from Lilly is the donanemab. So it is interesting that there's that they're only really looking at, they only see one of these things working in this assay.
Yeah, and this is informative this is informative for the future in clinical trials, because you can imagine that even if the hypothesis is correct that you need to start treating early, if you pick the wrong antibody, then you won't have efficacy again. And then, so let me introduce a second assay here, which is an assay that has really permeated the entire field of neurodegeneration. So for example, Parkinson's disease and other diseases also have been thought to follow a similar paradigm to what we've talked about with Alzheimer's, where there's one protein that misfolds and aggregates and then causes pathology. And Alzheimer's has kind of served as a paradigm for the study of neurodegenerative diseases, because a lot of the genetics and pathology were seen kind of earlier than the Parkinson's and other diseases. And so a lot of the assays that have been developed for Alzheimer's have been really spread out throughout all of neurology. And this assay is something that you see in many papers for all neurodegenerative diseases. And what they do is they take the brain of a mouse with Alzheimer's pathology, or you can also take human brain from autopsy, and you mash it up into a homogenate, and then you can inject it into the brain of a naive mouse or a recipient mouse. And then that recipient mouse will have pathology that is theoretically seeded from the injected homogenate. And this, actually, I kind of lied. This is actually not really from Alzheimer's literature. This is from the prion disease literature, where you can inject a prion and then it creates subsequent prions in the new mouse.
This concept's actually, I think, quite important, because it does transcend Alzheimer's, in that one of the commonalities of these diseases is that there's identified proteins that are misfolded, or there's too much being made. And so one can think of this as that the other big or main hallmark of these diseases is that you only get them when you're old, right? So most of the people that are going to get neurodegenerative disorders in this case are in their 60s and older. And so age is a risk factor, and these proteins often cause these pathologies. And all the so Parkinson's, all the main neurodegenerative disorders have these hallmark pathologies. And what's clear is that in some cases, you could have a brain that has all these pathologies in them, even this alpha-synuclein that causes Parkinson's disease. But they're focal. They also only affect certain parts of the brain. So if we could figure out a way of clearing out these proteins or getting the brain to get rid of them in a normal way, you could actually potentially solve a lot of these diseases, not just one. But again, coming back to the controversy, it's still not clear which form of these proteins is the real toxic culprit. And in this case, what Ori's saying is, what mentioned is that you have these intermediates, these oligomers, and it's thought that they could then seed subsequent pathology down the road.
And so we can draw on it. Yeah, go ahead.
Is Alzheimer's transmissible with brain extracts like a prion?
Yeah. So there's the data for this is a little bit complicated and is in the context of Creutzfeldt-Jakob disease, which is one of the prion diseases. And what they see is that in a patient who had some sort of transmission of Creutzfeldt-Jakob prion, they can also have transmission of amyloid-β and amyloid plaques. And it's like small case series. The evidence for this is actually better in Parkinson's disease, because in Parkinson's disease, therapy that was used for a long time was injection of fetal dopamine neurons into the brain of patients with Parkinson's, and then that essentially replenishes dopamine levels, and then you have symptomatic benefit. But in autopsies of patients who receive these fetal dopamine neurons, you can actually see alpha-synuclein pathology in the fetal neurons, which is presumably transferred from the host brain into the transplant.
And this question of spread is also interesting because, again, most of these diseases start off in one part of the brain, but by the time you get to the end point, where you're the end of the disease, the pathology is spread out from these focal points, and so there is this idea that there is transmission within the brain. This is actually a question my lab is interested in studying, and we're studying at the moment. But just to make sure, though, that people understand, prion disease is something different because we know the mechanism of the transmission, and that's caused by the misfolded protein being able to turn normal protein it comes into contact with into that same conformation, and that conformation is toxic. Here, it's slightly different, where you have these oligomers that then themselves can sort of influence their surroundings and end up causing pathology or seeding pathology, but it's not exactly the same mechanism, and they're not in a conformation that will change the conformation of these other proteins. So the way they spread is not clear, and they're not, even if there is some evidence for transmission from one person to another in this sort of weird way, they're nowhere near as effective as prions, where if you get some of the prion in your brain, that's it, you're done, because that's sort of a cascade that's uncontrollable. So
a lot of this prion idea has come from autopsy data, which is cross-sectional and has shown that essentially if you take time from diagnosis as the amount of time that the disease has been present, you can see that patients who have their time from diagnosis is very short, only have pathology in some parts of the brain. And then if you take a patient that has a longer time from diagnosis, they have pathology in that region and other regions that are also generally synaptically connected. So that is also a putative mechanism for transmission. The problem is, of course, that these are not longitudinal studies. These are cross-sectional studies. And the time from diagnosis is not a true time scale, right? Because who knows what it means that you were diagnosed at a certain point. The pathology could have been going on for much longer in your brain, but you were able to compensate and not have cognitive deficits. So these are kind of really hand-wavy ideas, but there's some circumstantial evidence to support them. I'll just tell you quickly, in one of my neurology residency interviews, the first question that one of the scientists asked me was, do you believe in the prion hypothesis in Alzheimer's? And I was just like, was like, I have a very nuanced answer to that. I mean, I was like, I guess I'm not going to go there. But anyway.
Well, even regular prions, of course, the Stanley Prusner, who sort of coined this term, was a pariah for a long time because he had this idea that it's a protein that's causing this infectious disease, not a virus. And he was, it took a long time for the field to be like, oh, well, maybe he's right. And of course, then he won the Nobel Prize. But it's sort of this typical ideas in science where you go against dogma and you can have this infectious disease that's protein based, not viral pathogenic based pathogen.
So the reason that this experiment is so interesting is that it gets at this infectious point. So Vincent, if we think about Koch's postulates, right, you can isolate the organism, but then you need to be able to show that if you take an organism from one animal that has a disease and you give it to another animal, then you can transmit the disease. So in the first part of this experiment, they measure the amount of A-beta. So it's like measuring the amount of bacteria in a sample. But in this experiment where they seed subsequent mice, they're actually showing whether the number of essentially infectious particles is changed. So what they do is they take a mouse, they treat it with aducanumab, and then they take the brain from that mouse and inject it into a new mouse. And if aducanumab is truly clearing pathogenic species, then the recipient mouse should have less pathology. In the recipient mouse, there's no exposure to the antibody. And they show actually in what I would argue is the most dramatic effects of any of the experiments that there's really reduced transmission of these species. So that's pretty strong evidence that aducanumab treatment, before there's even neuropathologic evidence of disease, is able to prevent subsequent pathology. So then they kind of, well, maybe you guys have other opinions about this, but they kind of then go on a series of experiments where they try to characterize what the pathogenic species is that they think that they're removing. And I was overall fairly unsatisfied with, I didn't really get an answer. And I think in the discussion, they acknowledged that they really don't know what is causing the disease, because you could imagine that it could be a monomeric species or an oligomeric species that they're clearing before they actually have true plaques on histology that's preventing disease. And they do this through, I think, really something that we can skip, but they don't really come to an answer unless you guys read something differently than I did.
No, I agree. I mean, well, what they do show is that there's sort of a distinction between some of these antibodies. So most of these antibodies they tested only really bind to the monomer versus the sort of seeds that they're looking at, or these aggregates, whereas aducanumab does recognize the larger assemblies. But in terms of sort of the, actual culprit, this has been the problem in the field for so long, in that every so often there's a paper that comes out that says, we found the oligomer that's bad. And but in reality, the evidence for that is sort of really convoluted and not direct. So yeah, not clear, but it's a hard problem to really figure out, I would say.
I mean, it's a very, very difficult problem, because you're essentially taking a non-pure species. You're essentially having to do purification and then characterization of proteins that are inherently difficult to characterize because they can multimerize and have these large hydrophobic surfaces. So it's very challenging. So to sum up the paper at the end, they then ask the question of whether they've kind of shown that they prevented amyloid plaque deposition, but do they prevent the subsequent parts of the cascade, which are tau pathology and neurofibrillary tangles, and then also neuroinflammation, so microgliosis and kind of other aspects of other hallmarks of the pathology in the brain. And they show very, very kind of convincingly that treatment with aducanumab at this pre-pathologic kind of stage of disease can prevent not only amyloid plaque deposition, but also neuropathological kind of progression of disease to tau and microgliosis. I think one thing that's missing, and I think is obviously very difficult to show, is a behavioral effect. So all of this kind of is showing that there's changes in the way the brain is. But what you really want to know is, does this affect cognitive outcomes in the mice? And the reason that's important is not just because I am being difficult reviewer number 3, but because that's really the endpoint in a lot of the clinical trials. So if you have an idea that you want to test this hypothesis clinically, you want to show that this is actually having an effect on the cognitive symptoms that you'll measure in the clinical trial, and that will affect the patient's quality of life and all of these things. And they don't do that in the paper.
Yeah, I had to admit it's kind of surprising that they weren't asked to do that, or they got away with not including it. Because these assays to look at learning memory, like the water maze and fear conditioning, are fairly routine now. I mean, labs that are not specialists in behavior can kind of do them, or at least you could find a collaborator pretty easily. So I agree. I mean, I would have liked to see that as an endpoint as well.
And I think that the other thing is that these mice are pretty sick. So I guess that makes it somewhat hard to show a phenotype, because you decrease the pathology by a little bit, and then the phenotype is it's a ceiling effect. But on the other hand, the pathologies or the phenotype is so strong that it should be an easy assay to see a difference between the two different controls. Yeah.
I don't think that's a problem. I mean, most of these animal models of Alzheimer's have been characterized with these standard behavioral tests, and you start to see. So let's say you start to see the first signs of pathology 6 months of age. By 1 year of age, you're definitely getting alterations in memory and plasticity, synaptic plasticity markers. So yeah, totally doable.
The other fascinating thing is that in these experiments, if you're getting tissue from a mouse that's 18 months old, it's very easy to run them in a behavioral task before you sacrifice them. So that it's not. So one limitation to these experiments is that it's quite expensive to age the mice for a long time. But if you're already doing it, it's very easy just to do the behavioral task. So I think that kind of sums up the paper. I think, in some ways, I guess. When I initially read this paper, I was surprised that this set of data hadn't been published or hadn't really been shown already, because in kind of discussions about these clinical trials, everyone talks about the fact that, maybe we're treating too late, or maybe we're treating the wrong A-beta species. And I assume that assumption came from data that earlier treatment actually had a disease-modifying effect. But it turns out that this is really one of the first demonstrations of this, and I think is really exciting because it gives an alternative explanation for why these trials could have failed.
Right. Now, playing devil's advocate, though, there's been some of the major controversy in the field has also been the fact that these mouse models are very artificial, because you often take a human mutation, or you're overexpressing a human protein in the mouse brain. Mice normally don't get Alzheimer's or dementia. And so you're driving artificially this pathology. And if you're trying to find therapeutics to these artificial constructs or these artificial models, how relevant is that to the human disease? Because in most of these cases, as we talked about, the cause of the Alzheimer's is not clear. It's not based on a mutation. This idiopathic Alzheimer's, which is like 80 to 90% of the cases, we don't really know what causes it. And so that's been the biggest sort of worry, is that sure, you get the pathology that looks very similar in the mouse and very similar to the human cases where there are no mutations. But if we solve the pathology problem, is that still going to really alleviate the most common form of the disease?
Yeah, I mean, I guess I'll play devil's advocate to the devil's advocate, but I think you have to start from somewhere. And if there's this is the best lead you have. And the question is, how much money as a kind of in the larger sphere of the biomedical enterprise do you dump into this hypothesis? And the question is there a trial that you could design that would give you rock solid evidence? And I think that there are trials like that are ongoing where they have, so the best way to test this would be to do it in a genetically determined population of Alzheimer's patients, so patients with an APP mutation or other dominantly inherited Alzheimer's disease. And those trials are ongoing.
Right.
So The problem is that it's harder to generate those cohorts and to have the numbers, but that's really the kind of the question of whether the genetic mouse models are relevant to idiopathic human disease.
Yeah, they've started. There's this one village or a couple of villages in Colombia where more than half the population have these dominant mutations, and so one of the trials that's ongoing has been targeting that population, and I agree. It's going to be very interesting to see, what the outcomes are. But I think this paper does highlight, as you said, that not all these antibodies are combating or targeting the same thing. So, are those antibodies that we're using out there the best ones?
Right, and part of that is also that when you see classes of drugs, a lot of times approval for one member of this class of drugs. So the first antibody that gets approved will then make it easier, or essentially make the standard of evidence be lower. The standard of evidence that's needed for approval for the second in that class will be lower. And I think that the point is that actually is the wrong way to approach this, because these are not really all in the same mechanism of action. They don't all have the same pharmacokinetic properties. So to say that if Biogen's antibody is approved, the Eli Lilly antibody should be approved more readily is the wrong way to do that.
But isn't one of the implications that you'd have to treat patients much earlier than we are, right? And in the context of a clinical trial, how do you do that, right? You have to treat people for ages, no?
Yep. Right. And the other problem is that the follow-up for this, is just outrageous, because you have to follow them for 5 years, or arguably even before they start converting into Alzheimer's disease, and then years for the progression of the disease. So these are hugely expensive trials. And then the second part is that the PET imaging, first of all, is not very sensitive, but also a lot of the ligands are only for the plaques. They're not for the monomers or oligomers that are the pathogenic species. So in some ways, if we treat early, we still won't have assays for target engagement. We're still going to be stuck with kind of cognitive tests and measures of pathology that won't manifest for years and years later. So it's really a tough spot. And I think that's why a lot of work is going into measuring levels in the CSF or in the blood, that you could have a kind of an easier way to measure the pathology.
Wow. So basically you take a population and you treat people, you give them the therapy, and then you just observe them for 5-10 years and see what happens, right?
Right.
Well, right. And then if you're in the drug company, you want to have it so that you're giving it repeatedly, so that they have to come back for more. So it's not even one dose. Some of them are
I can see where these genetically
yeah.
predisposed populations, like the one in Colombia, that they're kind of attractive because a lot of people are going to develop, but you don't know if that's, representative of the rest of the population. That's the problem.
Of course, yeah.
Right.
Wow.
Yeah.
Yep. That's tough.
Well, I don't know if you remember Uri, but there's this idea that herpes infections might be. Now they actually have ongoing trials for acyclovir.
A gram of it.
Yeah. And I actually think that's interesting. There's my take on this is that that any brain injury, whether it's from a virus, a bacteria, a fungus, a hit in the head, all of that's going to predispose you to dementia and Alzheimer's if it causes an inflammatory response. This is my pet hypothesis. And but the fact that that you could get HSV, and we always think that HSV is latent in the brain and it's not doing anything bad, is wrong. Clearly that's probably doing something, and that's a worry, because so many people have HSV, and so I think it's definitely worth looking into.
Well, then there's two competing issues. One is that the evidence isn't super strong, and I think that there's I don't know enough about the sequencing kind of methodology, but there are papers that are coming out all the time saying that essentially all that is artifact and noise in the initial papers that showed HSV sequences. But on the other hand, acyclovir is so cheap, and if you can have them, it's the question, and relatively benign. So what's the downside to trying something? It's a complicated question.
The problem is the the way they enroll patients is to do a PET scan and look for early onset, right? When it may be too late.
Yeah. Right. And that's what this paper is pointing out.
But the other thing we should mention is that, and there have been some papers suggesting that amyloid is part of an antiviral response, right?
Yeah.
Right.
Yeah. And that's, so that's a conversation that I think is very interesting, partly because there's also been a ton of work, like literally thousands of papers, trying to figure out what this whole cascade is actually useful for normally. So why do you have this peptide that's being cleaved? Why is it hung out for so long, despite the fact that it's probably not a good thing for the brain? And so, yeah, one hypothesis that sort of emerged was that maybe beta amyloid, the peptide itself, is an pathogenic peptide, the microbial, that it kills bacteria or viruses. And yeah, the evidence for that's also not convincing, or not super convincing, but maybe, I don't know.
I guess so. So the other thing that complicates all of this is that there are small molecule inhibitors of the proteins that cleave the APP into A beta and the other subunits, and those inhibitors are actually have safety concerns because they lead to cognitive impairment on their own. Right. So theoretically, actually, A beta, or the processing of APP, is required for normal cognition.
Right. There is this idea that beta amyloid itself is also regulating synaptic function, and is actually involved in this homeostatic form of plasticity, where you want to sort of dampen the amount of excitation in the brain. And the problem with Alzheimer's is that because you get too much of this peptide, that actually the initial dysfunction is synaptic dysfunction. You get a loss of synapses, and the loss of synapses seems to be the best correlate for the dementia itself, not necessarily the cell death. So we're actually interested in that aspect too. So what is this beta amyloid really doing in the brain?
So I think that for me, this is what's exciting about science, is that, we're saying all these hypotheses, but we're really looking for data to support them. And this is what science is. You can put out a hypothesis, you can put out experiments, and then over time, the truth will kind of sort itself out, or whatever is the truth in science. But it might take years, and unfortunately, people are suffering. So you don't want the process. You don't want the process to be shorter, but it may not be able to be shorter because that's how it works.
Right.
So should we go to emails and kind of get through a bunch of them? Now we have a backlog, I feel like.
Yeah, let's get through some. We have one really long one with multi-parts, and then a bunch from one person and then one from another. So let's start with that first one from Dave, who writes, I've been listening to the entire This Weekend catalog since March, when a friend who is a nurse turned me onto it. Having a background in biomedical science, prior to going into the private sector, I find I can reasonably follow all but TWiN. For some reason, the nervous system stayed outside of something I had any reasonable grasp on, like a foreign language only partly learned. Anyway, I've been mountain biking during the pandemic quite a bit, as have many folks out here in California. With the rise in bikers, there has also been a rise in injuries. Unfortunately, some serious neck and spine injuries in our area. I've witnessed 3 concussions that have raised my curiosity as to what is going on. In all 3 incidents, there was a fall and head strike, and all the riders had helmets on. For the first 5 or so minutes, the riders reported that they are okay, could tell you what happened, and were coherent. They could even finish riding down the trail. And then after 5 to 10 minutes, it's like someone took a magnet to the hard drive. They suddenly don't know where they are. They no longer know what happened, having just explained the crash minutes before. This seeming loss of short-term memory lasted a few hours in each case. Further, during this time, the riders would repeatedly ask what had happened. The incident would be explained, and then a few minutes later, they would ask again what had happened, to the point it almost seemed like they were joking. They weren't. In each case, we explained what had happened back to them at least 15 times. After a few hours of this, they were normal. So what's going on in their brains? Best to all of you, and I really do appreciate the podcast.
Yeah, I mean that's common, right? That's the sort of hallmark of these concussions, that initially it's sort of like, I don't know, I think maybe Ori has better analogies, but I see it as when you, let's say you have a cut and the cut is initially, it's not too bad, but then if it gets, if it starts bleeding too much or ultimately get infected, it's way worse than the initial injury. In the brain, initially when you have the physical trauma to the brain, it's almost like the whole head, the whole brain gets shook around. It takes a bit of time for the bad cascade to initiate, where things are getting released, bad peptides are being released, bad inflammatory responses are starting, and all of that then interferes with the consolidation of memory. And that's of course why you have amnesia after a bad concussion. And usually it's retrograde in that a really bad amnesia, you basically, a really bad concussion, you can't remember for hours before the incident, because there's sort of that consolidation process, which takes hours, is interfered with. And of course, making new memories can also often be affected. And that's why concussions are really debilitating in some cases, because people observe that they can't remember things as well for a few months at least after that concussion.
I don't have much to add. Concussions are out of my scope.
All right. So Margie had written a long email a while ago about a paper, and she has a few questions about it, but I don't think we can answer those because we haven't read it, and maybe we'll go over, but maybe we can move to the fourth question. Ori, you see that one? Fourth question about authorship. Maybe you could read that.
This is about authorship. From the SAS et al. Articles website, author information, these authors contributed equally. Why is it important to make note of that? And why are only those two mentioned when there are more authors listed? So in biology, I guess I should clarify, authorship is in kind of two competing orders of contribution. So if you are a young scientist like me, who's a graduate student or a postdoc, you contribute the most of the work, you drive the project, you will be the first author. Whereas if you're like Jason or Vincent and you're more senior and have different responsibilities, so you raise the money, you coordinate logistics, you provide mentorship, you then would be the last author, which designates that you are the primary kind of senior mentor author. And then for a number of different reasons, both because science has become kind of more multidisciplinary, that people graduate and then someone picks up and finishes the project, you can have two people who contribute equivalently to the same work. And then those people need to be designated in some way on the paper as equal contributors. And unfortunately, because of the way reward in science works, if the only thing that matters in some way on my CV is the number of first author papers that I have. And part of that is the number of co-first author papers. So if I contributed equally to someone else, I want there to be a demarcation of that on the paper so I can get equal credit to that. I think that is the answer to the question. Well, I guess the second part is that then there are other authors that contributed maybe reagents or a small set of experiments that involved less time and less effort or less intellectual input that then are other authors. So you want to demarcate that the second name on the paper contributed more than them or equally to the first author, but still contributed a lot.
Yeah, it's a little messy. I mean, I think this is actually you often see Twitter debates about this co-authorship idea, partly because, as already mentioned, the first author is stamped in gold. The first author in citations is the one that's mentioned usually, and so even if you have the star next to your name, the order does matter. And unfortunately, that means that there's really no such thing as co-authorship, because if it was, then you should be allowed to swap the name order, right? You should, if you really contribute equally, but that's a no-no. I don't recommend doing that. But it comes down to how you assess the science, and so it's all about credit assignment. and who actually drove the project. And this can get murky when you have a paper that may have, years of work and many authors. And it's tricky because now you get these quite detailed acknowledgement sections of who did the work and who contributed what. And that's good. And that's better than what it used to be, but it's still, I think, a little murky in terms of really trying to decide, this person should be co-author or not. And then, of course, the senior side of things, the sort of PI, the person that's running the project, for them, for their CVs, the corresponding author is the person who mostly gets the credit for that work and that project. But sometimes when you have collaborations where you have a number of labs, that you could see the stars at the back end of the paper as well, because you have contributions that are equal from each lab. And a lot of these sort of big science projects that you're seeing now have that. And I think that, and that's a conversation for another time, where I think the way we assess science and the way we credit people in science could do with a revamp.
And that's actually, so what is mentioned here is actually not the crazy, is nowhere near the craziest set of author attributions that I've seen. So you can have 5 co-first authors. This is a little bit of a weird story, but on the paper that I've mentioned a couple times on TWiN that we have in revision now. So I kind of started the project. A graduate student that's in the lab now is continuing it and has finished the experiments, but I'm still doing the writing and a lot of the analysis. So initially we were co-first. We each had a star, and Avery had his name first. But then more work happened, and essentially now I'm the last author because I also raised some money for it. But we still have our stars. So it's it's so complicated.
Yeah,
anyway.
I had some issues during my training where I had co-authorship, and sometimes I felt like it was fair, sometimes it didn't, and it all comes out in the wash. But it is tricky.
When I was a student, none of this was an issue because there was less collaboration. You typically did your own project, and first author was clear. And now the problem is their groups are huge, and everyone contributes a different big portion.
Right.
So that's where you have this issue, and it never did happen before. So it's a manifestation of the modern way of doing science in a way, But I agree, it's not perfect. We need to work it out.
In physics, that's why the author list is alphabetical.
Right. Yep.
Yeah. They can have hundreds of authors, right?
Right.
Yeah.
Let's do one more. Let's skip down to the next one from Dave. Jason, can you read that? It starts with good day.
Let's see how far. Oh, yeah. Good day, and thank you for your very twin-formational podcast. A comment was made that if an olfactory neuron was destroyed, it wouldn't regenerate. Although this is often true, olfactory neurons can and do regenerate even following transection. This paper reports on mice, but it has also been established in humans, and we can post the link. I've always thought that since chemoreception has been involving longest of the senses, suggesting a simpler structure with more time for incremental upgrades, that repair after injury has developed more than other senses. Dr. Dave Jackson. So it's true. The olfactory bulb, the olfactory neurons can be regenerated. It's one of the few parts of the brain where there's constant neurogenesis, so new neurons being made, and it is pretty unique. In humans, though, that olfactory sense, that olfactory bulb part of the brain is very small, and it's not clear, how well regeneration occurs. But it's true that that's one of the few parts of the brain that can regenerate.
So, Uri, what do you think about these people who still haven't recovered sense of smell after COVID? Think they're going to get it back?
I don't know. There was I'm involved in this retrospective chart study on all the neurological manifestations of patients at Columbia who have had COVID. And there have been a couple of patients where not only do they now recover smell, but they start to have this thing called parosmia, where they smell different things. They, for example, they smell pizza and they smell Chinese food. They actually, there is pizza, but they smell Chinese. Huh. And so now there are people doing cognitive training. So essentially, you get the pizza, and then you can do this like cognitive behavioral therapy to convince yourself that it is pizza, and then you retrain the brain. So there are lots of, I don't know enough, but it's crazy.
Interesting.
Yeah.
All right. Well, we have more, but we'll do them next time. And wrap up TWiN 14. Show notes are at microbe.tv/TWiN. Send your questions, suggestions, comments, twin@microbe.tv. And if you like what we do, go over to microbe.tv/contribute. Support us. We appreciate it. Ori Lieberman is at the Columbia University Irving Medical Center. Ori Lieberman on Twitter. Thanks, Ori.
Thanks, Vincent. This was fun.
I think this is our first of the new year, right? First one?
First of the new year. Happy New Year.
First Happy of the New Year. First of the decade.
Doesn't seem like that because this year is already dragged on. Unbelievable long.
Jason Shepard's at University of Utah. Jason Synaptic on Twitter. Thanks, Jason.
Thanks, Vincent. Thanks, Rory.
Thanks, Jason.
I'm Vincent Racaniello. I'm at virology.blog. And you have been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.